Good afternoon, everyone here. Welcome to the first of our virtual talks this weekend. It gives me great pleasure to introduce Professor Gene Robinson. And Gene is somebody that I have wanted to have at the convention for a number of years, but his busy schedule hasn't been able to fit us in. So it's great he's here tonight. Gene Robinson really needs very little introduction. He did his PhD at Cornell and has been at the University of Illinois for a number of years and has been involved in some very important projects, including the honeybee genome sequencing project. But here I think he's going to tie a lot of threads together with the talk entitled Me to We with Bees, Searching for the Genetic Roots of Social Life. So Professor Gene Robinson.
Thank you very much for the kind introduction. And good afternoon, good evening to all of you. It's a pleasure to be here. I would, of course, prefer to be there with you, but I'm really delighted and honored to be able to speak with you this evening. So yeah, I will be talking about some research in my lab that we have conducted over the years that really get at the question of how do you make a social bee? How does that happen, both in terms of brain mechanisms, and at the very end of the talk, a little bit about the evolutionary perspectives. To set the stage for that, let me turn to a sweeping view of all of life with a couple of key points highlighted for you and circled in red. So the emergence of the individual is something that happened during evolution, and you can see it happened about 2 billion years ago. Before that, life was a giant, how shall we say it, just a giant communal affair, literally, with microbes sharing genes, horizontal transfer of genes, and really no sense of group or individual, just one undifferentiated set of masses of life. This, of course, is the theory of RNA-based life, origin of life, which I'm proud to say the namesake of the institute that I'm honored to direct, the Carl R. Woese Institute for Genomic Biology. Carl Woese was one of the architects of this early thinking. But my point today is not to dwell on the microbes, but to set the stage that from that mass emerged the concept of individuality, the individual. And ever since then, the individual, of course, has enjoyed striking success and great radiations in the eukaryotes, and plants and animals are, of course, the most well-known. And this sets the stage for the conversation we're going to have this evening, which is that social life has evolved from individual life. And social life is complicated and involves many interactions. It involves a variety of adaptations that have had to happen. I'll also say that by way of introduction, that social life is relatively rare. And premier examples of social life are mammalian societies and insect societies, of which, of course, honeybees are a preeminent insect society. So with that, let's launch into what we're actually going to be talking about tonight. I'm going to tell you 3 stories. One, cocaine makes bees dance more. I'll talk about that. That will be the second story. Maybe I should have kept the third story because that might keep you here in your chairs wondering what that's all about. We'll start by talking about the ideas that the bee brain actually works a lot like the fish brain, the mouse brain, and even our own brain, and show you a little bit about common molecular building blocks of sociality. And then the third story is the idea that sick bees stay home from work. In a more serious vein, this is the outline of the talk. I'll talk about how you build a social brain, how you tune a social brain for altruism with respect to food, collecting food, and how you tune a social brain for altruism with respect to hygiene. Okay, with that, let's launch into the first part of the talk. The basic question you see in front of you, and that is, do brain responses to social information involve evolutionary conserved mechanisms? So a social animal needs to be able to respond to cues from the environment. Many of these cues in the context of a social animal are themselves social, that is, coming from other animals, from conspecifics, members of one's group, members of one's society. They need to be recognized, perceived, processed, and then they lead to flexible behavior. So we asked the question of whether brain responses to social information have similarities at the molecular level with considering mice, stickleback fish, and honeybees. The group picture you see on the bottom is our team, multidisciplinary team. The Woese Institute for Genomic Biology is a team science institute. Individuals work together in teams, multidisciplinary teams. You see our team here, Saurabh Sinha, right here, is a computer scientist. Dave Zhao is a statistical genomicist. Lisa Stubbs studied neurogenetics of mice. Allison Bell, neurogenetics, evolution of stickleback fish, and then my lab representing the honeybee. So we developed, based on natural behavior, we developed three paradigms that we wanted to study. Two paradigms across the three species: social challenge and social opportunity. Social challenge plays out differently in different species with different natural history, but at its core, there's a similarity. There's an intrusion into a territorial space. And for bees, of course, this occurs as disturbances at the colony entrance. Social opportunity reflects opportunities for reproduction, to gain reproductive advantage, to increase care. And so we focused on the care concept, parental care in the context of the stickleback fish, alloparental care for mice, and queen larval nursing. Of course, as you know, worker bees don't reproduce. And so to rear a younger sister into a queen is a tremendous social opportunity. It's as good as it gets for workers. So this is the natural behavior. For honeybees, we developed lab assays for colony intrusion based on beautiful work that has been done over the years, starting with Mike Breed, where you can simulate the responses of bees to an intruder at the entrance by creating small groups in the laboratory in plastic dishes, letting them form a colony group feeling, so to speak, and then introducing unrelated individuals and then scoring the aggressive behavior. We use the same general approach to, instead of an intruder, to put a queen larva in to look at bees that are responding to that opportunity. What we did then is, after developing the assays and exposing the individuals to a social challenge or a social opportunity, we would wait a certain amount of time and then perform dissections on the brain, particular brain regions implicated in social responsiveness, the appropriate brain regions in each species, and then measure brain gene expression. Measure how the brain responds at the molecular level to a social challenge or a social opportunity. The statistics of this are complicated, both to measure how to identify which genes are expressed at a different level, either increased, more activity, or decreased, less activity within the species in response to a social challenge, and then also identifying which genes are responding in a common way across the three species. But this slide just kind of gives a general overview. I won't take you through the general statistics. DEGs refer to differentially expressed genes. FDR, it means a very stringent statistical level, because we are comparing the expression of 15 to 20,000 genes. We're looking at an assay, looking at technology that can measure the activity of all the genes in the brain. And so for the honeybee, for the mouse, and the stickleback, this work requires having a very high quality sequence of the genome, and you heard Norman talk about the Honeybee Genome Project earlier, and this is what enables us to be able to do the work in this way, having a sequenced honeybee genome. And the take-home message is in the title right here, and that is when you perform these experiments, perform the complex statistical analyses, you do see that the same genes representing some of the same processes in the brain respond to a social challenge, whether you're a honeybee, a mouse, or a stickleback. And even though the nature of the social challenge, as I showed you before, is different. Let me give you just a little flavor for that with respect to the honeybee. And that is to point out that we see these genes that are responding differently to the social challenge. And the question is, what's going on? What are they doing? So when you present bees with an intruder, you get an aggressive response. That mimics what happens, as I think many of you know, at the hive entrance when there's a robber bee trying to get in, or an ant, or a bear attacking the hive. You provoke an aggressive response. So here what we did is we dropped an intruder bee into the dish that you see on the top right, and provoked an aggressive response. We saw changes in the activity of literally thousands of genes, many of them common to fish and mice, as I said, and a good number of the genes were involved in brain metabolism. That is, of course, genes code for proteins, and the proteins, many of them, not all of them, and not nearly enough yet. We're still in a very early stage of functional analysis in genomics in general, but many that we do know showed that they are involved in changing brain metabolism. So the question is, does this change in brain metabolism that we see in all three species, that is accompanied by an increase in aggressive behavior, is there a causal relationship to those two things, those changes in gene expression and change in behavior, or is that a correlation that means something else? So we tested that with bees, and we took two drugs that affect brain metabolism. We injected these into the brain, and we measured the effect on aggression. The dotted line that you see here at the point, at the one value, is the control value for what aggression we see when we put a related bee into a dish, not a foreign bee. And then hopefully what you can see is a statistically significant increase in aggression when we alter brain metabolism in the way suggested by the changes in gene activity, indicating that this has a causal relationship. These were experiments that were performed by former postdocs Hongmei Li Biarley and Claire Richoff, who now run their own labs at other parts of the United States. So what does this kind of result tell us? It's very important to frame this properly. What this tells us is not that bees are fish or fish are mice and so forth. It tells us that there are building blocks that evolution will use and reuse over and over again, independently, to build social brains. Now, why do I assert that? Take a look at the diagram that you see here. And here we have what is thought to be the last common ancestor of the vertebrates and the invertebrates. It lived some 600 million years ago. Very primitive organism, no centralized brain yet, no social life as far as we know, raising then these very interesting questions. Because when you look over here, we have the evolution of insect societies, and we have over here the evolution of vertebrate societies, including our own. If the last common ancestor shows no obvious level of social life, then the simplest conclusion is that these societies have evolved independently. One did not lead to the other. They evolved independently. Having said that, what we are finding using the latest tools in genomics is that even with these independent evolutions, nature is making use of common building blocks repeatedly to build social brains. So that is a profound new insight, and it leads to questions along the lines of why. Why are some particular molecular processes necessary, so to speak, in order to build a social brain? So that's a really important area. But let me push this even further. So we talked about bees, fish, and mice. Now I want to talk about some provocative connections in the way the bee brain works and the way our own brain works. This came about as a serendipitous discovery when the postdoc who was working on this project, Dr. Haggai Spiegler, who now has his own lab at the Volcani Center in Israel, when he was working on this, he was collecting bees using the challenge assay, as I said, a foreign bee, and that's depicted here for you schematically, red. He also used the same general setup, as I mentioned, with a queen larva, got the bees responsive to the queen larva. Those were the bees responding to social opportunity. And he ran those assays, collected those bees for the analyses that I was just telling you about. However, Haggai noted that there were some bees, when he ran the challenge assay, that did not respond to the challenge assay. There were some bees, when he ran the opportunity assay, that did not respond to the opportunity assay. And so it made us sit down and think about this. Are there bees that are less responsive or unresponsive to social cues? So the first thing that Haggai did was repeat these analyses, but now instead of working on a different group for the challenge and a different group for the opportunity, the same bees were given exposure to both of these stimuli and repeatedly. We wanted to see what can you do? Maybe you just were not attuned to it one time, but giving them repeated exposure, would there be any bees that would not respond? And the answer was yes. There were some bees that were unresponsive to multiple stimuli. And you see this graphed for you on the behavior diagram. And what you see measures social responsiveness. And let's just stick with the three groups that we talked about: those that responded to the social challenge in bright red, those that responded to the social opportunity in blue, and those that did not respond to either stimulus, and those are in green. So you can see there were some bees, about 10 to 15% in every one of our trials, with large numbers of bees, as you can see, hundreds, that did not respond to either analysis, to either stimulus. The next thing we did, so are they a thing? Are there really socially unresponsive bees? We have some behavioral data here. To support this further, we asked whether there are gene expression profiles that are distinct in unresponsive bees. And you see those data on the right. For those of you attuned to statistics, this is a principal component analysis, a rather high-level treatment. So let me just go under the hood for this for a second and say, when we compare brain gene activity of these three groups, so-called guards responding to the challenge, nurses responding to the opportunity, and unresponsive bees, when we look at compared gene expression, we find again about 1,100 genes differentially active in those three groups. What you see here is a way of just grouping that variation to kind of get a feel for the results. And I think you will agree, if you look at this, that the red dots and triangles tend to cluster together, the blue dots and triangles cluster together, and the green dots cluster together, more or less, meaning that these are three distinct groups. There are 1,100 genes differentially expressed, and those patterns are so consistent. Each of those dots or triangles are individuals, and they are coming from two different colonies. They are so consistent that these individuals tend to cluster together. So it really does look like there are a group of bees that are unresponsive. But, you know, does that make sense to have unresponsive bees in a colony? Maybe we're just not measuring things correctly. Maybe there are other possibilities. So let's push this a little further. To do this, we turn to our automated behavioral monitoring system developed by a postdoc in the lab, Tim Gernot, who was a computer science student at the University of Leipzig. And hopefully you can see these barcodes. And here's just a little video just showing how they look when the bees are interacting. What it is a very high quality, high powered camera, a special set of infrared lights. I think many of you know bees cannot see under infrared. And then we have a one-sided glass wall observation hive. The camera snaps an image of the observation hive one image every 2 seconds. There's only one side to that comb, and so all the bees, in theory, need to be present and accounted for. Machine vision identifies each bee, so every single bee in the colony is individually marked with the barcode. Machine vision identifies them, and I can tell you that in each frame, the program is able to detect between 80 and 90%. Sometimes one bee is on top of another bee, as you well know, and so you can't identify them. But we're talking about 24/7 monitoring, one image every, excuse me, 2 seconds. So terabytes of data and high levels of observation. The behavior of interest is the one circled for you right here. This is the first behavior that we have started to study, and this is trophallaxis, the sharing of liquid that's primarily food, but also containing a provocative cocktail of nucleic acids, signaling molecules, some beautiful work going on in the laboratory of Adria Bart in Switzerland. That is elucidating some of the chemistry of the trophallaxis fluid, telling us that it's not just food. There's a lot of food that exchanged in these fluids, but there are other compounds as well. And this behavior, known as trophallaxis, has long been thought of as the social glue of the insect society. Ants do it, bees do it, a variety of other species of wasps, social wasps do it, and it's constant exchange of materials. And so we focused on that. So machine vision identifies the individuals. Machine learning identifies this particular behavior as trophallaxis. By the way, this method will be featured in the third part of the talk, so you'll want to keep it in mind, and I'll go very fast at that point when I refer to this system. So we were looking then at the trophallaxis social network. And so that's another form of social behavior. And we then mapped out the levels of sociality of the individuals. And what we found was very interesting. There is a great deal of individual variation. First, let me point to you the queen, showing large numbers of interaction partners and large numbers of interactions. That is our positive control. That's our ground truth. We expect that to happen because, as you well know, worker bees are very attractive to their queen and they are interacting with her a lot. But now change your focus to the other bees. So this is just a pictorial sort of, called a hairball network. Size of the circles represent the number of interactions. There are some bees that have large numbers of interactions, really rivaling the queen, numbers of interactions and interaction partners. And then there's some, such as the yellow bee, very few interactions and very few interaction partners, reminiscent of what you might expect from a socially unresponsive bee in the assays that I showed you before. We tested this and found indeed that that is the case. This now brings the work, these two assays together. This was work that was spearheaded by postdoc Adam Hamilton in the lab. And on the left, what you see is that kind of a diagram, that general network diagram. On the right, we graph out the number of interactions for particular groups. We looked at foragers, we looked at nurses, we looked at aggressive bees, looked at bees that respond to multiple stimuli, and then the non-responders, the socially unresponsive bees, if you remember the green group from the previous assay. And then we graph the number of interactions. And what you can see is that the non-responders, the brown or the grayish blob on the right, have the statistically significantly lower numbers of interactions in the dish, and lower number of, or I should say, lower number of interactions in the observation hive, and they are the ones that are socially unresponsive. So it really does look like socially unresponsive bees are a thing. To then get to the main point of this part of the talk, we asked whether the brain gene expression profiles, the gene activity that goes on in the brain of the socially unresponsive bees, in any way has similarity to people on the autism spectrum, because one aspect of the autism spectrum behavioral profile is decreased social responsiveness. We did this with two data sets from the human biomedical literature. The first one is a data set that identifies gene variants. So now we're switching gears from RNA, which is how you measure gene activity, to the actual sequence itself, the DNA. And so we compared genes that have variation, genetic variation in their DNA sequence that is associated with being on the autism spectrum, identified that list. We had our list of genes from the honeybee, from the socially unresponsive bees, derived from gene activity, not DNA, but RNA. And we asked if there were any significant overlaps. And the answer is yes. The yellow horizontal axis there shows that there was a significant overlap for the top rated genes in the Safari GSM database, that is the genes most closely associated with human autism spectrum. Then we did a second type of analysis for gene expression, that's on the bottom of the slide, and that was post-mortem brain gene expression. You cannot get real-time measures of human brain gene expression because it's a destructive assay. So to measure gene expression in the brain in humans, that comes from autopsies. And again, we found for our socially unresponsive genes, we found an overlap with the genes that show differential expression based in humans based on post-mortem results. In other words, there's evolutionary conservation of autism-related genes in honeybees. And I indicate, for those of you that are interested, some of the mechanisms, heat shock proteins, some neural signaling pathways, some very well-established pathways of neural activity. So this is a provocative finding, and we took pains to do as many different control type analyses that we could. So we took the B gene set, the socially unresponsive gene set, and compared it to gene sets from other human mental conditions, and we did not find an overlap. Conversely, we took the autism gene sets that I just showed you, the two gene sets, compared that with other gene sets from honeybees that distinguish other groups of honeybees, guards or foragers and so forth, and we did not find an overlap. The only statistically significant overlap we found was what I showed you, the socially unresponsive genes, honeybee gene list, and the genes that are associated with human autism. So one more time to go over this, because it's really important now that we're talking about this particular finding. Bees are not little humans. Humans are not big bees. And I also want to say that I am not in any way speaking glibly about human autism spectrum disorder. It's complex. There are other phenotypes, traits associated with it besides social unresponsiveness. We are simply asking whether there may be building blocks associated with the social brain that extend from deepest roots of evolution, play out through independent evolutions of social brains, including our own, and can be then implicated in cases where the social brains do not operate in a neurotypical way. So I've talked about how you build a social brain in terms of identifying common building blocks. Now that we have a social brain, let's talk about how it gets tuned for altruism, and then in the final part of the talk, how it gets tuned to keep healthy. So I think it would be safe to say that, loosely speaking, at least, many people can say there's some addiction to sweets, some more, some less, but there's that sort of thing. How about honeybees? Could you say that they are addicted to sweets as well? How do we use that concept of addiction when we're moving from humans to a social insect? Well, let's explore that. And the reason why we want to explore that is because altruism is at the root of a successful society. Individuals that are performing activities for the better of other individuals in their group. And we see many examples of it across the insect societies. We have dance language, which we're going to talk about in a moment. We see the manufacture, a conversion of nectar to honey involving group activity, cooperation to build nest mounds that are unbelievable. This is not a toy, this is a real giraffe. These are termites, tiny, about the size of a honeybee, and they're building these giant mounds in Africa. We have one of the world's oldest examples of farmers, the leafcutter ants, collect leaves, not to eat the leaves, but to use them as a substrate to grow fungus, which is their food. And then, of course, as in all societies, and this is particularly poignant to be talking about this at this point in time, we have warfare, such as these tournament ants, where you have the red ones and the black ones engaging in warfare and raids on each other's colonies and so forth. And so all of these are based on a division of labor and a social organization that has at its core altruistic elements. So let's focus in on food. And here is the motivating insight. When a solitary insect finds good food, she eats more. Simple. However, when a honeybee finds good food out at a flower patch, she doesn't eat more. She pretty much takes the same size of the load, but what does change in her behavior is she dances more. So we have a me to we shift from personal behavior, eating more, to a social behavior, communicating the good news to other individuals. So I want to take you through some explorations, molecular explorations, and neuroanatomical explanations that connect these two behaviors, the selfish one of eating and the altruistic, the social one of dancing. So the dance language, I think many of you are well familiar with it. It communicates distance, direction, and the value using a series of encoded movements and sounds. We took advantage of a great method that was developed in Manyan Srinivasan's lab that tricks bees to think they have flown further than they have. This allowed us to perform some highly controlled experiments by seeing whether we could test whether neurochemical systems that are involved in motivating selfish behavior, eating, are also involved in regulating dance behavior. And what this arrangement that you see here, this is an outdoor flight chamber. So screen, the bees can't get out. We can put food here that has the substance that we want to expose the bees to in their food. And we know they'll get it because that's their only food source. We have a glass wall observation hive here for monitoring dance behavior. And we have this tunnel that we train the bees through to get to the food source. This was important because this allowed us to monitor exactly the distance that the bees thought they were flying, because the tunnel tricks them to think they've flown further than they have. So we could get high quality dances to a distance that we were very sure of, to be able to look at effects not only on the probability of dancing, but on the quality of the dance. And what we found, this is now the work of Andy Barron, who has his own lab at Macquarie University in Sydney, Australia, is that octopamine makes the bees dance more. We chose octopamine because when you give it to individual solitary insects, it makes them eat more. So on the y-axis, you see the likelihood of coming back from a feeding experience at a feeding station and dancing. And then we have two controls. We have bees that had the syrup, sugar syrup with nothing added to it. We have the solvent that was used to dissolve octopamine, and then we have octopamine and the solvent. You'll notice how low the level of dancing was with the control bees. We did this on purpose. You can manipulate this by giving bees a low quality of food so that we would have a low baseline and be able to detect an upswing in dance behavior. And you can see the bees are more than 3 times more likely to dance if they've been treated with octopamine. So we see the me to we switch, but how very clearly. But to push this a little further into the way that I introduced this, having to do with addiction, we needed to get closer to the territory of addiction. And so we chose the substance cocaine to see if cocaine also makes bees dance more. We chose cocaine because obviously it is highly addictive, and also cocaine actually is chemically related to octopamine. So we had strong expectation that cocaine would have the same effect as octopamine. It still was a good experiment because you never know. If there had been a surprise and we would not see this effect, it would really call into question this conceptual framework that I've been sharing with you about how the reward system can be manipulated and tuned to motivate social behavior. So same kind of experiment. The 3 groups are repeated as before. Cocaine has the same kind of effect. And furthermore, when you treat with cocaine and a substance that blocks the receptors, you decrease the dance behavior. So from a pharmacological point of view, a nice tight set of experiments that makes the point that cocaine makes bees dance more. We've also looked at this in the context of gene expression and in the context of neuroanatomy. So we did another experiment. This is now the work of Matt McNeil, who works at IDT Biotech Company, and Karen Kapheim, who has her own lab at Utah State University. And we did the me and we experiments. We had bees, if you see here in panel B, we had individual bees starved overnight, given a droplet of sucrose syrup, a classic, some of you recognize this as the proboscis extension assay, that all the beautiful work of Randolph Menzel and many others over the years have been used to study honeybee learning. So you give a bee is by herself, she's been starved, you give her a droplet of food, she eats the droplet of food. So it goes from the crop, which is where the food is stored temporarily when bees are foraging. In this case, goes from the crop, the foregut, into the midgut. It's actually eaten. It's a selfish behavior. We also did a dance experiment, trained bees to go to a feeder, and then identified those that came back and danced, removed them from the hive, and then measured their gene expression. And what we found was that there were many genes that show changes in activity when a bee received a meal. Panel B. So those are the red circles. The blue circles show that a subset of those genes, same genes, subset of those genes also were responsive to dance. And in fact, we clustered them. There was one cluster, dance 1, dance 2, dance 3, dance 4, that represent coherent molecular activity, the same kinds of biological processes. So we have a me to we story here at the level of gene expression as well. Dance responsive genes are embedded in the food responsive gene set. The panel on the right extends this to neuroanatomy. We used a particular gene that is known as an early immediate gene, a gene that responds quickly to changes in environmental conditions. And we did the same experiment. We did a me experiment. We did a we experiment. And this time, rather than measuring all the genes, we used that gene in such a way that it would give a visual marking of where in the brain it was lighting up, where in the brain it was turning on. And the red areas, again, are all the areas that light up, that respond when a bee has had a meal, the me case. The blue represent areas of the brain that light up when there was dance. And as you can see, the green areas represent the overlap of those two. So there was an overlap of brain regions that lit up both in response to food and in response to dance. So it leaves us with these kinds of questions. Do bees dance more because, just to use the cocaine experiment, cocaine makes them like their food more? Does it enhance their pleasure? Is it affecting the reward system? Some of the regions in the brain that light up in the honeybee brain are responsive to reward because they're turning on in response to food. So what we're saying is that dance is a rewarding stimulus, consistent with the idea that the reward system has been shaped to promote altruistic behavior. Now, stepping back, I think, for those of you that have ever tried to teach children to grow up to be good citizens and kind people and responsive people, this might sound familiar. And that is, we try to shape the behavior of children to encourage them to take pleasure from doing good deeds, from doing the right thing. And the reward system, we know in humans, is a very pliable system, a very flexible system. And for some people, they get deep pleasure from listening to opera singers. From other people, they get deep pleasure from going to a football game. And if you do brain imaging studies, you will find that whatever it is that gives a person deep pleasure lights up the same parts of the brain. So the reward system is very flexible and has been adapted to take on a variety of connections between stimuli. Sadly, that kind of flexibility also leads to addiction, where drugs can hijack the reward system. So I think it's not far-fetched to talk about social evolution hijacking the reward system to promote altruistic behavior. Okay, the last part of the talk is how we get a sort of an idea of how we tune social brains for altruism for another purpose, and that is for hygiene, a subject on our minds all too much in these last couple of years, especially with respect to viruses. So bees, of course, are afflicted with a variety of viruses, as are all organisms. And we're going to talk about one, Israeli acute paralysis virus, IAPV. It was originally associated with colony collapse disorder. It infects all life stages of bees. It's a pretty serious virus. It's transmitted from individual to individual, and it can cause death. The work I'll be talking about is a collaboration with the laboratory of Amy Toth, a former postdoc in my lab who has a laboratory at Iowa State University, and Adam Dolezal, a new hire to our department here at the University of Illinois. So we wanted to ask about altruism from a different perspective. How do bees handle the threat of disease spread? One of the costs of living in a dense society is that disease can spread. And again, a lesson that we have learned all too well over the past couple of years during the pandemic. So what kind of adaptations, what kind of responses might bees have to this threat? So we can think about this in a couple of ways. First of all, you can't just talk about the host, you have to think about the pathogen as well. What's in it for the pathogen? How does the pathogen evolve to spread more? and how does it manipulate host behavior. So if disease spread within a honeybee colony is controlled, is decreased, one mechanism could be what we call the stay at home from work. So you feel sick, you are encouraged not to go to work so as to minimize the spread. So you're minimizing your social connections in response to a perception of having a virus infection. That would be a host advantage. That would be good for the bees. On the other hand, what's a sneeze? A sneeze is a pathogen manipulating our behavior to spread itself. And so we might see behaviors that increase the advantage of the pathogen. How has social evolution handled all this for bees, and how does it handle it with respect to the pathogen, in this case, IPV? We turn to our barcode system. As I mentioned before, every bee in the colony has a barcode. We use colonies of about 1,500 individuals. We again focus on trophallaxis. And what we found was that infected bees interact less. So it's the equivalent of staying home from work. They're interacting less with members of their colony when they are infected. That is, I should have said, we infected some bees artificially with a known amount of virus, and we tracked their spread. And then we had colonies set up that were exactly the same genetics, same size, same food, but they did not have the virus. And then we compared their social interactions. You see the number of trophallaxis interactions per bee per hour, again, around the clock for 1 week. And you can see that in all 3 colonies, they interacted less. You might have thought, well, maybe they interacted less because they were sick. So they just couldn't move. They couldn't interact with other individuals. No, this seems to be more sophisticated because infected bees move more than the control bees did. So they had more opportunities to infect each other, to exchange fluids with each other. But as you saw in the previous graph, they did it less. So that gives an advantage to the bees over the pathogen. There's less spread within a colony, as there could be, based on this behavior, this tuning of the social network. However, that's not the whole story. And again, we know this all too well from the pandemic. It's not just local spread, but spread over larger geographical scales. So in this case for honeybees, what about disease spread between colonies? And here, if you're a bee enthusiast and you were happy to see that the bees had the advantage, here you're not going to be feeling so good. Infected bees forage more. So we had our barcoding, we have the ability to monitor foraging behavior continuously. Infected bees do forage more. They take more trips per hour, as you can see. And they are attacked less by guards. So what's the connection between foraging more and attacked less by guards? Well, the more bees forage, the more likely they are to drift. Especially take an apiary like this, very close, colonies are close together. They're not marked in any distinct way at all. I think many of you know there's a lot of drifting that goes on. So if bees are foraging more, the stage is set for them to be able to drift more into other colonies. And when they do that, they are attacked less. So here we have 2 controls. We have bees infected with a virus, the purple or the magenta. We have light blue, bees that were just given sugar. And then we have a treatment that stimulates the immune system very generally, but is not the same as the particular virus. And as you can see, the bees with the virus are attacked less than those that are stressed with a general immune stressor or those that just get sugar. We saw this both at colony experiments. We saw this also in a version of the intruder assay that I showed you before. So if they're attacked less by the guard bees, that means they're more likely to get in to the colony. And in fact, they also are fed more by the guard bees, increasing their chances of being able to make it into a colony and spread, therefore, the disease. Infected bees smell differently. Here's another one of these principal component type analyses where you measure many, in this case, not genes, but many compounds on the cuticle of the bees, and then cluster the individuals. And you can see that the control bees, the red bees, have the name, the technical name for the immune stimulant, and then the blue bees are the virus ones. You can see they all cluster separately, those three groups. Infected bees smell differently. We presume that's why they're treated differently at the colony. So we have a complex story here. We have less social interaction in the hive, less spread within the hive. A nice example of a me-to-we adaptation, advantage B. However, the virus is not sitting pat, not standing pat. There's coevolution. There's pressure on the virus to be a better virus, to spread more. And so there are effects to increase foraging, which likely will increase drifting. That's an advantage for the virus. And then once those bees are at the site of a foreign colony, they are more likely to get in, meaning it's more possible for them to be able to spread. So let me finish up with this slide here. So we've talked about the building blocks to build a social brain. We talked then about how you tune a social brain that's responding similarly to challenges and opportunities, but take it to the next level, to not just responding as individuals, which is what those experiments were, but to really shape altruistic behavior, dance language, and the social network within the hive. So we've talked about all of those changes that go on. And the question that this work here addresses is, how does that happen? How do you evolve higher levels of altruism? How do you evolve these changes? It's commonly known, or it's commonly thought of, dating back to Darwin, that this is a process, an evolutionary process, where evolution acts on the group rather than the individual. In the case of social insects, we call it colony level selection. And many people have talked about this, have shown behavioral traits that could only be explained in that way, such as dance language. Individual bees don't dance, so that must be selection, evolutionary selection at the whole society level. I just want to end by showing that now in the genomic era, where we have the ability to study these kinds of things at the genome level, we see a very nice example of colony level selection. That comes from an entirely different system, and that is in Puerto Rico. A former student of mine, Professor Tua Garay, discovered that the African honeybees that arrived on the island of Puerto Rico started off being very aggressive, just like they are everywhere in the New World, but within 10 years, evolved to be much less aggressive. That is a swarm that Professor Garay's student, Miguel, is holding. And for those of you who know bees, you can do this with European bees. This is not recommended with African bees, but here's someone doing it with African bees to highlight the fact that they have lost a lot of their aggression. So what you see then is a highly technical slide here, and this is the last data slide. And this is called a Manhattan plot, where what you do is you sequence the genomes of hundreds of individual bees, and then you look for variation in the genome sequence at specific spots, single nucleotide spots that correlate with variation in behavior. And so typically, this is called, for those of you that are interested in this field, a genome-wide association study. So this might be done for heart disease susceptibility. You have a group of individuals, you measure the degree of heart disease, you sequence their genomes, and you look for correlates, single spots on the genome, single nucleotide spots on the genome, where variation exists repeatedly in many individuals correlated with a particular disease state. So we ran that kind of analysis for the Puerto Rican population of honeybees, and we took bees that, this is the individual here, we took bees that were either highly aggressive, soldier bees that responded to a threat, came out stinging, or foragers, not as aggressive. During the disturbance, they kept going in and out of the hive to forage. We sequenced the genomes of those individuals, about 300, and then we looked for variation in the genome that correlated with variation in behavior. The dotted line that you see is the threshold that of the strength of the association that you would need to call it a significant association. And I think you will see that the area above the dotted line contains no spots. There were no spots. There were no places on the genome that correlated with differences in individual behavior, whether you were an aggressive scout or a less aggressive forager. However, we took those bees from 10 different colonies, and when we shifted the assay, instead of asking whether you were a scout, whether you were a soldier or a forager, we asked whether you came from colony 10 or colony 3 or colony 1, and we knew the aggressiveness of each colony. Now, all of a sudden, see all these dots here? And these arrows point to the strongest ones, super strong associations between variation in the genome and variation now in colony behavior. So the colony phenotype is associated with the DNA variation, and is then therefore manifesting Darwin's idea now at the level of the genome. And I'll just say and finish, this was the work of Ariana Avalos, who's now at a USDA, a postdoc in the lab, who's now at the US Department of Agriculture Bee Research Facility in Louisiana in the US. And a graduate student in the lab has gone on to extend this to find the changes in gene regulatory networks in the brains of individual bees that are tuned and by the colony level selection. So to finish up, how do you make a honeybee? Well, you take a ground plan of solitary bees, you layer on top of that molecular universals of the social brain, you layer on top of that group selection, colony level selection, and you run the experiment for a few 100 million years, and you'll get honeybees. That's a very quick summary of the talk. I also want to highlight how we are able to do this kind of research, in part because of the powerful interplay between basic and applied research with respect to honeybees, the historic strength of apiculture that teaches those of us that do research on honeybees so much about the natural history of bees and the ability to manipulate bees to be able to do these kinds of experiments. So let me close by highlighting some current and former lab members. I tried to highlight their contributions along the way, some collaborators for this work, and then some of the funding agencies associated with this. And I will be happy to take your questions. Thank you very much.
Thank you very much, Gene, for a very thought-provoking talk. Where you started with the mice and the sticklebacks, has there been other research done on those creatures that parallels the work that you've done on honeybees?
Yes, there has been. So in the lab of Alison Bell, some of the genes that show common responses are being studied. There's genome manipulation that's possible to test this in a functional way, and the same for mouse. So yes, the work does continue. And I talked about both, that we've studied both social challenge and social opportunity and covered very little of the social opportunities. So there's that whole side of things. Yeah, one of the big questions in this field relates to why are there particular molecular pathways that form these building blocks? So that's where some of the research is going.
Thank you. The question relating to the bees with the Israeli acute paralysis virus and the fact that more of them get let into hives. Do we know whether there are other examples with other viruses, for example, chronic bee paralysis virus? Do we know whether the same works for those?
We know for DWV the same holds. In fact, it probably has more effect. And we've studied DWV with this in mind, and it affects the activity of many genes, and there's an overlap of the genes that are affected by DWV and the genes that change during behavioral maturation. So it makes bees forage more. We saw this behaviorally in the data I presented today, and we also presented this in terms of the gene expression changes in a paper published a few years ago.
Thank you. Is it cause and effect that your name is Gene and you work on genes?
Yeah, I'll have to defer to my late mother on that. That was her decision.
So does anyone know if there's an advantage to society for the fact that these genes have been conserved through so many levels in terms of their effects on sociality?
No, we do not know. It is a frontier question of why some molecular pathways are required for a social brain, and repeatedly. We do not know that.
So going back to early on, where you had your guard bees, your nurse bees, and your unresponsive bees, we know that in honeybee colony, bees take it in turns to do those jobs as they age. So they start off as nurse bees and then become guard bees later. So was it that these individual bees were always unresponsive, or was it at a certain stage in their going through their age polyethism?
Mm-hmm. Yeah, that's a great question, because whenever you assert not or then it's like, well, how many different ways did you test them to be able to say that? In this particular case, our experimental design was to work with bees only at one age, but all at the same age. So one of the features of division of labor in honeybee colonies, it's very flexible. And so if you create a colony of bees all the same age, you get division of labor. You get foragers, meaning precocious development. You get bees that show normal development as nurses and so forth. So in the dish, that's the same thing. You kind of get a mini division of labor taking place. So all we can say was that at the age when we could find responsive bees as nurses and responsive bees as guards, we did not see these particular bees showing responsiveness.
So do you think that's because you've got different patrilines of bees in the colony that relate to different drones, that that there's something about the genetics of that particular group that they become the guards or the nurses, even though they're the same age?
I think it's both nature and nurture. So in our particular, so I'll give two answers. In the experiments that I showed today, we actually controlled for genetics partially. All the bees were the offspring of one queen and one drone. So they were more closely related, but as you well know, there's recombination in queens, and so they're not clones of each other. So they're genetically more similar to each other, suggesting that there are social dynamics involved, random factors that could be involved. But then my second answer is we have followed up on the study now together with a British collaborator, actually, Jen Cook from University of Birmingham, and we're doing the GWAS studies, that the kind of study I mentioned at the very end, looking at DNA variation associated with social responsiveness. And we do find it, to your point, we do find it. And we're finding it in, again, some very provocative genes associated with sociality in humans as well. And so we're working on that. And it just, it's another reason why the Bee Society is such a useful reflection or model of other societies. So it's entirely possible that there are small differences due to genetics that could then get magnified by social factors to give rise to bigger differences. So if you think of stimulus response, if there are bees that, so I'm calling them unresponsive, a more nuanced way to look at them might be less responsive. So if you have them operating in a colony and they're with other bees that are more responsive, those are the ones that are going to be the first ones to do something. They will decrease the stimulus level, so it's never going to cross the threshold where a less responsive bee is doing it. So you assay them under certain conditions and they're not responsive, but under other conditions, they might be. And for those of you that know the bee literature very well, you will know that bees that don't perform tasks have been seen before, and they've not been called socially unresponsive. They've been called reserve bees. Bees, not based on a whole lot of data, but very plausible. The idea being that, well, maybe they're keeping their powder dry and they're not expending themselves. And if a calamity hits a colony and stimulus levels go way up because there are fewer bees to do a job, you know, they might be there and they might well be responsive in that context.
That's fascinating, because my colleague Steve Martin did some modeling work some years ago, and you know, he showed that with a theoretical disease, you could actually take quite a large proportion of the population out before it really had any effect on the productivity of the colony. And right. You know, everyone assumes that honeybees are really busy all the time, but actually there are, if you watch an observation hive, there are bees that really don't appear to be doing anything, and that's fascinating.
Yes.
Right.
Yes.
I think we'd better sort of wrap it up. I think we have one question at the back there, Rinke. So Rinke was just saying that, of course, we have the eusocial insects, and you know, they're very different from the sticklebacks or whatever, but we do have naked mole rats. Is there anything we know about similarities between those and honeybees in the way their genetics works, the gene expression works?
Yeah, they are a fascinating group. And of course, we in the social insect world call them honorary social insects. They have not been studied as intensively from the genomic perspective. They are harder to rear. There are fewer colonies that are easily available. Again, this is my homage to beekeeping. So, you know, we have over 100 colonies. We can get sample sizes for our genomic work that other people would die for. You know, we just are able to do that. And naked mole rats are much more difficult. So there's been some very interesting hormonal work. And so common endocrine systems that are involved in regulating reproduction and reproductive maturation in vertebrates have been tweaked to give rise to queens and workers in naked mole rats, but to my knowledge, detailed genomic analyses have not been done.
It's fascinating that there is so much more still to do, even though we're beginning to get an understanding. Right, well, on behalf of everyone here, I would like to thank you very much for taking up your afternoon talking to us. It was you know, great to hear your talk, and I think it's been incredibly thought-provoking. So can we thank Gene again?
Great. Well, thank you very much. It was a pleasure to be here, and I was very glad to have the opportunity, and I would look forward to coming back in person.
Yes, hopefully we can get you over here in person at some point.
That'd be great.